So hello friends, so in this video I am going to discuss you what are the types of necrosis, and finally at the end I am going to show the histopathological structure of each of the types of necrosis. So please friends, so let's discuss what are the types of necrosis. The first type of necrosis is the coagulative necrosis, second one is the liquefactive necrosis, third one is the caseous necrosis, fourth one is the fat necrosis, and the fifth one is the fibrinous necrosis. All these are the 5 types of necrosis. So now I am going to discuss the histopathological structure of each of these types of necrosis. So friends, so from this part I am going to explain the histological structures of each types of necrosis. So let's begin with the histopathological structures of coagulative necrosis. So that you should know that this is the model showing the histopathological structure of coagulative necrosis. Now see that in this histopathological structure, see that, so here these are the normal tissues of the kidney or the normal renal tissue. So therefore that is viable renal tissues, that means all these tissues are alive. And these are the, so here that is the normal or dead, so here renal tissue, so therefore that is, you can see that necrotic tissues. Now see that in these normal tissues, that in each of these cells, that is nucleus is there, and in the nucleus that is a normal amount of cytoplasm is there, and also that is the outer boundary of the whole tissue is present, and along with that is the cell boundary of each of the cells is present. So this is a normal tissue. Now see that in a necrotic tissue, you can notice that is the nuclear content of each of these cells is not maintained, it is destroyed. And also on the basis of the destruction also, there are nuclear states can be called pycnosis and karyohexis. Now you can see that, and also that in the tissues also there is a cytoplasmic structure is also destroyed, and also in this tissue that is the pinkish eosinophilic content has increased as compared to the normal living tissue of the renal. So see that in the normal viable renal tissue, so this is the, so here that is a pinkish or the eosinophilic content, but in this necrotic tissue, the, so here pinkish eosinophilic content has increased too much. First you can also see that the nuclear content and the cytoplasmic content, so the nuclear content and the cytoplasmic contents of the cell have been destroyed, and in this, so here necrotic tissue only that is the outer cell, outer boundaries present or preserved. So therefore that it is like a ghost cell. So therefore that is the each of the cells of the necrotic tissue is known as ghost cell, because nucleus, cytoplasmic structures have been destroyed, only the, so here that is the cell membrane or the outer boundaries preserved, and each of these cells is like a ghost, so therefore ghost cell. And also that is pycnosis, karyohexis, these are the stages of the nuclear damage. So these are only informations about necrotic tissue. And also you can notice that in this histopathological structure of coagulative necrosis, so here see that is the living renal tissues are found, and also dead, so here necrotic tissues are also found, and at the junction of necrotic tissues and viable renal tissues, you can see that, so here that is the inflammatory cells are found, and these inflammatory cells are mostly see that, so here lymphocytes. So once again, what are the structures found? So see that is the viable renal tissues are found, and also necrotic tissues are also found, and here see that in each of the cells of this necrotic tissues, you can see that a nuclear content is not maintained, neither cytoplasmic content is maintained, only the outer portion of each of these cells is maintained, so therefore each of these cells is like a ghost. first thing. And you can see that, and on the basis of the nuclear damage, so here pycnosis and karyophysis can be said. And also you can see that this is a junction between the living tissues and necrotic tissues, and between these two junctions, so here inflammatory cells are present, and these inflammatory cells are mostly, so here lymphocytes. So these are the only informations that you should know. So see that what I've written about us. So here it is the identifying features. So here it is the only the cell outer boundary preserved, whereas the cytoplasmic and nuclear contents of the cell destroyed. So therefore these cells are known as ghost cells. Second, that necrotic cells, they have more eosinophilic content than non-necrotic cells. As you can see here that these are the non-necrotic cells or viable cells. So here you can notice the eosinophilic content in these normal cells, and also these are the necrotic cells. You can notice the eosinophilic content. So here that is more eosinophilic content in a cytoplasm than this. So that is a pinkish eosinophilic content. And also see that third. So here that is an inflammatory cell infiltrate present at the junction between normal and necrotic cells, and this inflammatory cell infiltrate contains many lymphocytes. And number 4, the cells also show pycnosis, karyophysis and karyolysis. So these are the here main identifying features of coagulative necrosis. Now so this is the next histological structure diagram, and this diagram so here shows the histopathological structure of liquefactive necrosis. Now see that, so in this histological structure of liquefactive necrosis, again so here see that, so these are the normal or the viable, so here tissues, viable tissues, and these are the so here or the dead or the necrotic tissues. Now again so here you can see that in this viable tissues, so here in each of these cells, so here that is a cell boundaries preserved, cytoplasmic content is there, and also nuclear content is there. But see that in this necrotic cells, so here see that is a nuclear content is destroyed, not a cytoplasmic content is present. And also you can see that that is all the so here cells they have, so here that is irregular cell boundary, as the cell boundary is also not preserved, it is also destroyed. And now here you can relate that I have written that is a liquid material. Why liquid material? Because that this necrotic so here cells they have lost their nuclear, cytoplasmic and the cell boundary contents, and after their destruction, and this so here necrotic cells converted into liquid like materials, so or into semi-solid materials. So therefore that is so here liquid materials I have written. So this is the first information. So here that is about a necrotic cells, that is a necrotic cells they have lost their nuclear, cytoplasmic and cell boundary content, and this necrotic cells so here converted into so liquid like materials, first thing. And you should also know that so here that these are the viable cells, normal cells or a non-necrotic tissues, and these are the necrotic tissues. So here see that between these viable and necrotic tissues, so here that is junction is present, and in this junction so here you can see that so here that is a granulocyte, a granulation tissue present like lymphocytes and also macrophages, and also blood capillaries are present. So just remember that at a junction between the viable tissues and necrotic tissues, so here lymphocytes, blood capillaries, and here that is the macrophages are present. So all these are the identifying features of liquefactive necrosis. And see what I've written. So here that is identifying features. The first one is a necrotic cells. They have lost their cell boundary, cytoplasmic and nuclear contents, and these dead necrotic cells get converted into semi-solid material and later into liquid material. And that type of necrosis is known as liquefactive necrosis, first. And second, you can also notice that between the normal and the necrotic tissues, so here that is a junction is present, and a junction a granulation tissue present, and this granulation tissue contains here lymphocytes, so here macrophages and blood capillaries. See what I have written there. Between the normal and necrotic tissue, so here granulation tissue is present, and that granulation tissue contains lymphocyte, blood capillaries and macrophages. So friends, so this is our third histopathological structure, and this, so here that is histopathological structure is about the, so here that is a caseous necrosis. Now see that in this histological structure also, so these are the, so here that is the normal, so here that is the lymphoid tissues, and here, and you also note that in the lymph nodes, so here lymphoid follicles are present, so these are the lymphoid follicles, and these are the viable lymphoid tissues. So all these are the viable lymphoid tissues, that means normal tissues. Now, and here see that, and these are the, so here that is the necrotic tissues, and this necrotic tissues converted into debris like structure, so therefore written, so here that is the granular debris. And here again this is the, so here junction between the, so here normal tissues and necrotic tissues, and this junction you can see there, so here that is different types of cells, epithelioid cells are present, giant cells are present, and these epithelioid cells and giant cells that together form, so here granulomatous, or that is granuloma. So all these are the informations. And here, so here how this granular debris form, so here see there in this also, so here cells, they have lost their cellular, nuclear and cytoplasmic all contents, and they got converted into granular debris. First thing about the formation of granular debris, and the second is about the junction. So here see that this is a junction between viable, so here lymphoid tissues and necrotic tissues, and this junction consists of giant cells and epithelioid cells, and these giant cells and epithelioid cells will do the formation of granulomatous in future. So these are identifying features. See that, so here identifying features, that is first, the necrotic cells have lost their nuclear content, cytoplasmic content and cell boundary, and these necrotic cells got converted into granular debris. And next point, between the junction of necrotic and normal tissues, the following cells are present which cause granulomatous inflammation. The first one is the giant cell, and the second one is the epithelioid cell, and this giant cell and epithelioid cells, they will do the granulomatous inflammation. So these are all the identifying features of, so here caseous necrosis. So friends, one thing that you should know that, now see that in coagulative necrosis between the normal tissues and necrotic tissues, so here a junction is present. So a junction is present between the normal tissues and necrotic tissues. So in this coagulative necrosis, normal tissues and necrotic tissues are seen. So just like in a liquefactive necrosis also, normal tissues, necrotic tissues seen, and a junction is also present between them. And in this caseous necrosis also like that, same thing. So here normal tissues present, necrotic tissues present, between them that is junction is present. Now, so friends, now I'm going to discuss you about the fat necrosis. So for fat necrosis, please see this diagram. So please see this diagram, and friends, I know that by seeing this diagram, so here you can see the label as cloudy appearance, and also so here about the inflammatory cells. Now see that that this cloudy appearance is about the necrotic cells. Now in this case, that is the necrotic cells, they have undergone or they have had fat deposition, and due to this fat deposition, that is necrotic cells, they are looking like clouds, so therefore cloudy appearance is seen. Now here you can notice that in this case, so here that is normal tissues or cells not seen, only necrotic cells seen, and also and along with necrotic cells, inflammatory cells. But here you should remember that in this case no normal cells seen, so therefore junction between normal and necrotic cells is also not present. Now, and third thing that you should note that in this diagram, so here that is the cloudy appearance, that means a necrotic cells, so here that is surrounded by a mixed inflammatory cells. Inflammatory cells like mixed inflammatory cells like neutrophils, basophils and eosinophils, and all these mesenchymal cells here surround the, so here that is a necrotic cells, that is a cloudy appearance cells. So all these points you should remember, and see that is what I have written there about. So here fat necrosis, so here that is the microscopic features first, that in the necrotic cells, so here fat deposition occurs, and due to which these necrotic cells have, that is, cloudy appearance. This is the first point. Second, see that no junction is present between normal and necrotic cells. And third thing, so necrotic cells, so are surrounded by mixed inflammatory cells, and these mixed inflammatory cells like, so neutrophils, basophils, and eosinophils, and these are the microscopic features of fat necrosis. So friends, now I'm going to discuss you about a fibrinoid necrosis. But before discussing fibrinoid necrosis, so here that is, please see the normal structures of the wall of a blood vessel. See that normally in the wall of a blood vessel that there are three layers present: tunica intima, tunica media, tunica externa. And please notice that in this normally, in this normal structure, that there are no inflammatory cells like neutrophils are present in the wall of blood vessels. So you should remember that normally in the wall of blood vessel no inflammatory cells like neutrophils are present, or no like that. So here hyaline like materials are present, so you should remember. Now this is a normal or key concept that you should remember. So now, so see that this is a diagram of fibrinoid necrosis. Now see that, so you can notice that in this fibrinoid necrosis in the wall of blood vessels, so here hyaline like material is deposited first thing. And second, see that, and this hyaline like material is surrounded by, so here that is neutrophils. So these are the only key points that you should remember, that in the blood vessel wall hyaline like materials present, and second, and this hyaline like material surrounded by neutrophils, and all these points I have mentioned. See that, so here the microscopic feature that in the vessel wall that is brightly eosinophilic hyaline like materials deposited, and second, and this hyaline like material surrounded by neutrophils. So here that is, so that's all about the histopathological structures of necrosis. So I hope that this video gives you a lot. So thank you.